Today, we will be focusing on the citric acid cycle and a biomolecule that specifically regulates it. But why do we care about the citric acid cycle? This cycle is the main source of energy for cells and is a part of a greater process of aerobic respiration. In fact, it is the most important metabolic pathway because it connects almost all of the individual pathways together. It gives us the electrons necessary for fueling oxidative phosphorylation. Here, energy is converted to ATP. One whole cycle of the citric acid cycle gives us 7 total products. These products include 1 GTP, 3 NADH with 2.5 ATP per molecule, 1 FADH2, which it has 1.5 ATP per molecule, and 2 CO2 molecules. So what does this really mean in terms of how we function on a day-to-day basis? Well, without the citric acid cycle, we couldn't breathe, move, keep our hearts beating, and even think. So it's a pretty important feature in our bodies, to say the least. Even if one of the intermediates is diverted, the entire citric acid cycle fails to function. So let's look at an important molecule that regulates this cycle. Isocitrate dehydrogenase is an enzyme that is specifically used in the third step of the cycle. CO2 is released from the isocitrate and catalyzes the oxidative decarboxylation of isocitrate. While this occurs, NAD+ is converted to NADH. In the fourth step, another molecule will take over alpha-ketoglutarate, but we are not focusing on that today. All the ATP molecules and electrons are produced through smaller steps throughout the citric acid cycle. This enzyme has an alpha-beta structure, and we can see 14 alpha helices and 18 beta sheets in its secondary structure. What is cool about this molecule is that the secondary structures are arranged in such a way to create sandwich structures. Due to this, the 2 active sites on the molecule face opposite directions. The active site is where the binding of NAD+ or NADP+ occurs, and additionally an Mg2+ or Ca2+. Although it may seem out of place, those metal ions seem to be important for catalysis. Here, isocitrate uses 8 amino acids like tyrosine, serine, asparagine, arginine, and tyrosine, and lysine to bind to the active site. So what does the mechanism and reaction look like? This is a spontaneous reaction which is driven by the reaction of NAD+ into NADH. Because isocitrate dehydrogenase has a very large change of free energy, it is a reversible reaction. The reaction begins with isocitrate dehydrogenase catalyzing the oxidative decarboxylation into alpha-ketoglutarate. Step by step, first, the hydrogen atoms are removed from the isocitrate, which is used in converting NAD+ to NADH, and an extra hydrogen atom. Here, an intermediate is formed called oxasuccinate. A carboxyl group is removed from the oxasuccinate, which is driven by another enzyme. A carbon dioxide molecule is produced and replaced with hydrogen. This forms the final product of this step, alpha-ketoglutarate, which is used in later steps of the citric acid cycle. Every single step of this cycle remains important, since without it, we would simply be unable to survive. If there are high levels of ATP, NADPH, or NADH, isocitrate dehydrogenase will not proceed with catalyzing the reaction. This whole process is carefully regulated so the amount of this enzyme is not completely depleted. This highlights the importance of the specific cycle as well as the individual enzyme.